All right. So you're welcome to my YouTube channel. My name is Bigba Bright, and I've always loved to do this. I talk science, I talk health science, and I talk genomic science. Um, I've always said that I'm not the only person who has something to say. I'm not a repository of knowledge. So today, I'm happy to be joined by what a very good friend from Kenya by name Ruth Nanjara. He'll be talking about um, human leukocyte antigen imputation accuracy. It's actually a work she has done, and she'll be telling us what all this is all about. And I want to take this opportunity for us to share some few knowledge about what all this HLA is all about. Ruth, you're welcome.
Thank you, Bright. Uh, and
Uh,
thank you for welcoming me to your channel as well. It's a
all
pleasure
right,
meeting you on here.
all right. Thank you very much. So, with to somebody who is hearing human leukocyte antigen imputation accuracy for the first time, I mean, what exactly does it mean? What does it entail?
Okay, so let's start from the basics. So if you hear about HLA or the human leukocyte antigen region, so what would you think this would be? So what is it's basically a locus on the human genome that is very essential for innate and adaptive immune responses. It is also important for organ transplants because I guess most of you have heard about organ rejections and things of this sort. So you have to match HLA types from the donor to the recipient. So HLA is key for that. It's also associated with um, diseases so we have autoimmune diseases like rheumatoid arthritis and ankylosing spondylitis, and also some infectious diseases like HIV. So this particular region of the human genome is very important. So when we talk about HLA imputation, what we mean is just trying to predict these HLA alleles. So you basically have your data, but you're trying to now, with no HLA alleles, so what you're trying to do is to predict them. So that is what HLA imputation would mean. So when we talk about HLA imputation accuracy, is when we're trying to now assess how accurate were these alleles predicted. So because we are using a statistical procedure to predict these alleles. So, the other reason would be that this region is highly variable, so it keeps changing. It's highly polymorphic. So you need to ensure that whatever alleles you get are accurate enough. So my work focused on just getting to understand, okay, if I'm going to use this particular tool or this particular software to impute HDLs, how accurate is this software? And for imputation, you also required to use a reference panel, so like something you can refer to. So what I also looked at was trying to just understand whether the different reference panels that are used, how accurate are they? Is it something that you can advocate or something that you can advise someone to use for their analysis? So basically, that is what my work revolved around. Back
Wow.
to you, Bright.
All right. So thank you very much. That's a very good summary. So I think you might have completed the work by now. I'm sure.
Mm-hmm.
Yeah. So I mean, is it a tool you have developed yourself, or you've tested it on a different tool, or people have done it and you were just trying to assess how what the tools function? Is it a tool you have
Well, it's
developed yourself?
okay. Thank you. So it's not. I didn't try the tools because I was trying to compare different tools. So I did not try the tools. So what I did was just writing a script, if I could say it in a layman's term, or a pipeline that can run the comparisons. So a pipeline that can compare now the different tools.
Okay. So,
Yes.
so all right. Thank you. So I normally say I like people say that scientists we speak in these big jargons. So I mean, to somebody who is not a scientist and maybe watching this video, I mean, how
Mhm.
useful is your work in the area of health, like, or in the
Yeah.
area of what is it? human
Yeah,
health?
sure. Sure. So I think I mentioned before that HLA alleles are linked to important human diseases. So things like rheumatoid arthritis. I'm sure some of you might have heard of this,
Yes.
or autoimmune diseases. Or I think everyone has heard about HIV. So
Okay.
HLA alleles are linked to HIV and or these other diseases that I have mentioned. So if I'm to explain this in a layman's term, I would just say that for you to be able to say that okay, this particular allele is associated with HIV, you have to be 100% sure, so that you do not say that okay, maybe HLA-B 27 is linked to HIV and it's actually not linked to it, because your outcome would serve for policies and implementations and things like that, because now people will be able to say okay, because this particular allele is linked to this particular disease, then what we can do is maybe we can edit this particular gene, or we can try to maybe things like CRISPR and do what something of the sort. So I think it's very important to ensure that whatever HLAs you say that are linked to a particular disease are accurate, and that is what my work was trying to come up with.
Oh, then what? Then that's interesting. Not to divert, I have read somewhere about the CCR5 gene, because you keep
Mhm.
mentioning the HIV, stuff. I've heard about this particular allele which is quite prevalent in somewhere in the East Africa, especially Kenya. I don't know. Have you heard about it? CCR5 gene, which what we have heard of is prevalence, or like it's being found in some people, which is making them resistant to HIV virus. I don't know what is your imputation. It might be an offset question, but have you heard about it?
Mhm. Yeah, so sure, I have heard about it, but you see, that is not HLA. It's not a HLA gene or a HLA allele, but I've heard about it.
Okay.
So with my work, if you're focusing on HLA alone, you are able to say okay, if someone has maybe HLA something, because HLA is denoted with 4 digits or 8 digits. So if someone has these digits of HLA alleles, then we can say that this person is maybe protected against HIV-1 or protected against HIV-2. And if someone has this other group of HLA alleles, then you can see that this person is at a higher risk of getting HIV compared to the other population. So you need to figure out okay, are these alleles linked to HIV, and are these alleles protective against HIV? So once you know that, you can then move ahead to look at your population, and then you can tell a person confidently that you are at a risk of getting HIV. So you should avoid this and this and this. Or you can tell them that you're protected against HIV, so you can do this and this and this. So I don't know whether that makes sense.
Okay. Yeah. What is that? That makes sense actually. So let me find out in terms of data. What is it? Did you, collect your own data or use publicly available data?
That's a very nice question. So I used publicly available data. You can easily access it online. So if anyone who wants to access it, it's Gambian data set. So you can write to me. I'll show you how to get it, but it's publicly available.
Publicly. So was it African origin? Was it the data?
Yeah. That was something I think I forgot to mention. So it's my project was focused on an African population. So yeah,
Okay.
it was an African data set.
Wow. So from here, what are you going to do with the project? You seem to have a very good findings, that you can move
Mhm.
ahead with, or something. Is it a work in progress? Is it something you want to pursue further, or something, a recommendation you want to make to government, to policymakers? From here, where are we going with
Mhm.
the work?
Well, so I might not proceed with the same work, but I'm supposed to start my PhD soon. So I might be working with a very different population and not the African population. but I'll still be working on HLA. So maybe what I would advocate for maybe people who would be interested with working with the African population in future would be that if you are to perform invitation, because there are various ways of getting HLA alleles. You can do sequencing, you can do HLA typing, but of course the easy one and the one that is cheaper is imputation. So for those who want to do imputation, I'd advise them to use HiBag. HiBag is a software. And also for the reference panel, they should use a reference panel that is at least population specific. So one that matches the target data set of interest, and it should also be larger in size. So with this, you are able to impute HL alleles more accurately. And once they have the HL alleles, they can now move forward to do association studies. So trying to link up, okay, I have these HL alleles, so how are they associated to these phenotypes of interest? So how are they associated to things like maybe cancer, um, things like maybe HIV, things like maybe arthritis, whatever phenotype they are interested in. Yeah.
All right. So thank you. My second but last question. So you notice that when people talk about alleles, genes, genomic science, it sounds like spirit. And people think they what they look like spirit. And so I don't know whether I'm putting you on the spot, but if you're talking about genomic science or you're talking about alleles, you're talking about genes, I mean, how do we break this down for the layman to understand? Genomic science is
Oh,
one of the things we are unable to convince policymakers, because when you're talking about genomics, people don't really get it, because it's like you're talking about some spirit hiding somewhere. You've been in
okay.
genomic
So let's.
science. Yeah, go
Yeah.
ahead.
I don't know whether this would help break it down, but when we are talking about genomic sciences, we basically talk about the genetic makeup of a human being. So what is it that makes you to be the person you are, or what is it that makes you susceptible to a particular disease? What is it that makes you maybe protected against a particular disease? You might have noticed that with the COVID outbreak, we didn't really have the heavy burden in Africa. So,
Yes.
but we see these burdens more lying on the with the European populations and the Asians, and yeah. So, what is it that helps us to be maybe immune against this kind of outbreaks? What is it that enables us to be immune against a particular group of diseases? So it's basically just the genetic makeup of of a person, and
Oh,
alleles would be now the alternative forms of a gene.
okay.
Yeah.
Oh, then what is it? That's a very good summary. And what is it? Thanks very much for that. And my very last question. So you notice that too many ladies don't like sciences. I
Uh-huh.
hope you can agree with me. If we are going to
Yeah,
take
yeah.
statistics, you will realize that very few ladies are into sciences, especially the area you are delving with. Is there some kind of
Mhm.
inspiration from you, or how do
Mhm.
you want to inspire other ladies to pick up from where you are now?
That's a very nice question. I'm actually in computational biology, bioinformatics.
Yes.
Um, so just to inspire women out there, or ladies, or
Yeah.
girls out here, is that this can be done. So it's not like it's meant for men alone or meant for a particular kind of species, but we can also do it. So if you are willing to put in the effort, if you're willing to put in the interest, and if you're willing to push beyond the bare minimum, then it's possible. Nothing is impossible. So long as you set your mind to do it, you will do it.
All right. All right. So, Ruth, thanks very much for making time to be around. And this is a very great pleasure and much more revealing to hear from you and to talk about um, the human leukocyte antigen imputation accuracy, at least for now, we have a very fair knowledge about it. Thanks very much for being around.
You're welcome. It was my pleasure.
And I promised I will get back to you to talk about other issues as and when they come up as far as genomic science is concerned.
Sure, no problem.
All right. So thank you.
Bye from Kenya.
All right. And it's also a very good afternoon in Ghana. It's just 1:38. Thanks very much. And to those who are watching out there, thanks very much. This is all that time will allow us for today. We promised to do it within 15 minutes, and I think we are done. I just spoke to Ruf Nanjala all the way from Kenya, and who has conducted a study into human leukocyte antigen imputation accuracy. And if you heard from what she said, what is it? A lot has been revealed. So watch the video over and over, and you can share it as well. Thanks very much. My name, as usual, is Biba Bright, and it's always a pleasure coming to you with science issues. Thank you. See you some other time.